Yo, 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 welcome to topic 7, gene linkage. So Morgan decided to investigate how different genes were inherited together in fruit flies. So Morgan crossed flies that differ in traits of body color and wing size. So the wild type, remember, this is the control, right? The normal of these traits are gray colored bodies and normal wings. The mutant type would be black bodies and vestigial wings. He crossed a dihybrid for these genes with a fly that had the mutant recessive traits. According to Mendel, the result should have been a 1 to 1 to 1 to 1 phenotypic ratio. 50% of the offspring were to be representing the parents, as seen here, and then the other 50% were supposed to have a recombinant phenotype, as seen here and here. And so that wasn't the case. The majority of the offspring had the parental phenotypes, and only a small portion had the recombinant phenotypes. So the scientists reasoned that the body color and wing size are usually inherited together in specific combinations because the genes are on the same chromosome. And this is called gene linkage. The alleles from linked genes are carried on the same chromosome, so they end up in the same gamete cell. For this reason, linked genes do not follow Mendel's law of independent assortment. Crossing over during meiosis allows some of the alleles on the same chromosome to separate, producing recombinant phenotypes, as seen here and here. Genes can be mapped on a chromosome by determining the recombination frequencies. So what does recombination frequency mean? It's the total percentage of recombinant phenotypes produced in the offspring from a cross between a dihybrid and a recessive individual. Remember, y'all, dihybrid means that it is heterozygous for both traits, and then recessive, this is a very specific type of cross. It's called a test cross. And so you should remember that term. So we have the formula here, the number of recombinants seen divided by the total offspring gets me that recombination frequency. And Morgan, using this cross, determined the recombinant frequency to be 17%. So this is an important fact that you guys must note. The farther apart the two genes are, okay, on the chromosome, the higher the probability that crossing over will occur between the two genes. Therefore, the higher the recombination frequency. Whereas if the genes were located closer together, the lower the percentage of crossing over will happen between the two, and therefore the lower recombination frequency. So a map unit is representing 1% recombination, simply the distance between genes, okay? Map units indicate relative distance and order, not precise locations of the genes, okay? So relative distance. Since Morgan's cross involving the body color and wing gene resulted in a recombination frequency of 17%, the two genes must be 17 map units apart on the same chromosome. Okay? So again, if we're given 3%, we know that we need to look at the highest percentage to determine the endpoints. So we can determine the order of where these genes would be located on a chromosome, and this is called chromosomal mapping. So again, we're given 3% here. The first step is to look at the biggest percent. That will tell us the end points. So A and C must be the end. Then we need to take this look at the second part or biggest percentage, and we see that it is A and B. We know that A must be as far away from B as possible. And finally, the smallest percent, and that makes sense. B and C are very close to each other. So again, B is located closer to C than A. We can use chi-squared to determine gene linkage. So the null hypothesis is indeed that there's no difference between all of those phenotypes, meaning a 1 to 1 to 1 to 1 ratio. And then if we reject this null hypothesis, we're stating that there are higher percentages for the parental phenotype versus the recombinant phenotypes, stating that, yes, these genes are most likely linked. Here's a reminder that the chi-squared and the chi-squared table are found on your AP Biology exam formula sheet, and you do not need to memorize it. Finally, let's take a look at it visually. If the genes are not linked, it means they are on separate chromosomes. And this is what it means to be on separate chromosomes. There are equal possibilities of getting all of these different recombinations. Okay? Whereas if the genes are linked, meaning they're on the same chromosome, again, the percentage of having the parental phenotypes will be much higher, higher than 25% than the recombinant gene or phenotypes. Okay? And those should occur at less than 25%. That's it for today. Have a good one.